In this lecture, we are going to look at anemias caused by defects of DNA metabolism. All right, so the etiology of these types of anemia is where impaired DNA metabolism impairs the production of all the dividing cells in the body. Sometimes we see a megaloblastic anemia. Megalo has mega at the end of it, and the root cause of this is impaired DNA synthesis. We see very large cells of the bone marrow and a macrocytic anemia. Usually, folate is essential to the production of thymidine nucleotides used in DNA production. So if we have a decrease of folate and also B12, we can start to see these types of megaloblastic anemia. Since some folate is catabolized, another source is needed, which is 5-methyl tetrahydrofolate, which releases its methyl group to form tetrahydrofolate with vitamin B12 as a cofactor. If folate or B12, as I just mentioned, is missing, thymidine nucleotide production for DNA synthesis is impaired. DNA replication process will be incomplete and cell division will halt, resulting in a cell lysis or apoptosis. So you need B12 and folate for the proper production of red cells. Some of the clinical findings we have. Megaloblastic anemia will develop when DNA synthesis and cell division is impaired due to lack of B12 or folate. These people experience fatigue, weakness, and shortness of breath. A vitamin B12 deficiency can cause neurological symptoms, memory loss, numbness, loss of balance. And a folate deficiency can cause cardiovascular disease. We see symptoms of depression, peripheral neuropathy, and psychosis. So what can cause some of these vitamin deficiencies? Well, a folate deficiency obviously can be from inadequate intake from foods, increased need, such as pregnancy and lactation. You can have an impaired absorption, so it's absorbed in the small intestine and only 50% of it is absorbed. We could have impaired usage or excessive loss, for example, through the kidney. In a B12 deficiency, we can also see it from an inadequate intake. We can see increased need, which occurs during pregnancy, lactation, and growth. We can see impaired absorption, in the case of pernicious anemia, which we'll learn about, or you can have a hereditary transcobalamin deficiency. So with the laboratory diagnosis, we have some screening tests. The CBC is always the most common one when we're looking at anemias. But with these types of anemias, we see pancytopenia, decreased hemoglobin and hematocrit, an MCV between 100 and 150, an increased RDW, and a MCB being increased. The MCHC would be normal. The blood smear would have oval macrocytes, and we'll see some pictures in a while, and hypersegmented neutrophils. We see dacrocytes, which are teardrop cells, fragments, and microspherocytes. We may see some nucleated RBCs, some Howell-Jolly bodies, and basophilic stippling. We can also see hypersegmentation of the neutrophils. The neutrophils may have lots of lobes inside of them. Oftentimes, an increased bilirubin and lactate dehydrogenase, which is due to the ineffective erythropoiesis and intramedullary hemolysis. So as we have increased cell turnover, the breakdown of the cells is going to increase the bilirubin. Increased cell turnover typically increases the lactate dehydrogenase, which is LDH. We have some more specific diagnostic tests. A bone marrow exam can help us identify developing RBCs and issues we may have with them. So if somebody has megaloblastic issues, this refers to the specific morphological changes, not just the overall size. The cytoplasm becomes more pink, and the nucleus lags behind in maturity. We see an ME ratio, which is myeloid to erythroid ratio of 1:1. The assays for folate, B12, and methylmalonic acid are also done. These are very sensitive markers for vitamin B12 deficiency, the methylmalonic acid. We can also do a gastric analysis. This is used to confirm achlorhydria, which is found in pernicious anemia. So that's a lack of hydrochloric acid in the digestive tract. One cause is pernicious anemia. So if somebody is lacking something called intrinsic factor, they can't get the B12 from their stomach into the bloodstream. We do antibody assays to look for these intrinsic factor antibodies or parietal cell antibodies. Parietal cells usually produce gastric acid, and intrinsic factor is a glycoprotein produced by the parietal cells. So we need those things present to get the B12 into the bloodstream. Another test that is done is the Schilling test. This one's used to distinguish malabsorption of vitamin B12 resulting from pernicious anemia from other types of malabsorption. This is done with a 24-hour urine collection. So the patient collects a urine for 24 hours, and the presence of a vitamin in the urine indicates that the patient is able to absorb vitamin B12. A second 24-hour collection is done with intrinsic factor. After they give the patient intrinsic factor, the vitamin should appear in the urine. This indicates that the intrinsic factor correlated the problem, and the problem was pernicious anemia. The vitamin does not appear, then there's another cause of the issue. Other things that can cause this, parasites. So we analyze the stool for eggs of a fish tapeworm called Diphyllobothrium latum, and they are very voracious eaters of vitamin B12. Isn't that pretty? A little tapeworm in the intestines. People actually swallow these to lose weight. The treatment for this would be to treat the specific vitamin deficiency. They'll give vitamin B12 intramuscularly if they need to bypass the need for intrinsic factor, and they can give folate orally.